So I want to thank Dr. Liu for the invitation. I've had a very eventful day. I was in Chicago last night giving a talk and drove back this morning, and it's quite, I was in traffic for about an hour. It's only a block from the hotel. It's just, there's everywhere. They've had a lot of rain. So it's a lot of flooding in all the intersections, and I drove my van through knee-deep water to get here in time. So I'm very glad to be back at Champaign-Urbana, where it's raining, but at least not too crazy, the roads. So I came up, when I was asked to do this talk, I tried to think of something that I thought would be of interest to you guys. I'm not very familiar with your department. I know Dr. Liu, but what bioengineers would be fascinated with what I do. So I tried to come up with something I thought was interesting, kind of a theme that we would have fun with. So we're going to talk about human milk oligosaccharides for the most part. So I'm going to start off with a little introduction about what are probiotics and prebiotics and human milk oligosaccharides. And then we'll talk about infant formula, which is where I first got interested in human milk oligosaccharides. And then some how these HMOs are metabolized by gut organisms. And then we'll talk about the synthesis of a particular HMO 2-ethol that we're very interested in, which is a collaboration with Young-Soo Jin, who's from our department, who came, I think, 3 weeks ago? Yeah. So, all right, so are you guys all familiar with probiotics? Does anybody know what probiotics are? Is that just a few people? Yogurt, right? That's why actually last night I was in Chicago, I was at a community event talking about yogurt the whole time. So we got yogurt on the brain today. But probiotics are defined as these live organisms, microorganisms which confer a health benefit when administered in adequate amounts. And actually in my class, this is very important. This is always a 3-point question because there's 3 parts. Live, right? Not dead, live. This is a problem because, for example, in yogurt, to get the organisms to survive during the 6 weeks, the shelf life of yogurt is not trivial. And so that's one issue that we deal with in food micro. It has to confer a health benefit, and so, it's not just not enough just to say, it's a bifidobacteria, there must be a health benefit. For a lot of the claims that people make, they have to actually be some evidence for that. And then the last part is definitely there's a dose, right? In fact, a lot of the yogurts that you buy might just say they have bifidobacteria inside. This is a little tidbit of information for you guys. If you look at your yogurt, if it says the strain, gives the strain designations. For example, it says Lactobacillus acidophilus NCFM. That's a guarantee that that strain is present in very high amounts, because the manufacturer of that strain will not allow the yogurt maker to claim that strain unless they know it's at high enough levels to be beneficial. Right? They don't want to be dragged down by people who just sprinkle it in, so it's very low abundance. And people think, that probiotics is garbage, right? When in fact, they're not meeting their expectation, right? So that's a critical part. It mostly consists of lactobacilli and bifidobacteria, which is perhaps my bias, but that's the 2 bacteria I study the most. And then we're interested in modulating intestinal microbiota to health-promoting population. And we'll talk about that some more. So we have lots of, these are some of these that are kind of dated because they don't exist anymore. Like the Vive, at least I assume that's how you say that cashew product. That was a breakfast cereal that actually had probiotics in them. The Attune bars you can get like at Strawberry Fields. We even have a horse probiotic there in the top corner. Juices, infant formula, which is part of why I get a lot of funding and do a lot of infant nutrition work, is a lot of interest in bifidobacteria and probiotics and infant formula. Even suppositories for infants, and supplements, and even things like the Dan Active or Activia yogurt, probably something a lot of you are familiar with, or other examples. So it's not just yogurt, but yogurt's definitely a major player. I love this slide because it's from Dr. Sanders' website, but this kind of lists all the different things that people have kind of, and this is not really the top focus of today, but just there are lots of different physiological or health aspects that probiotics have been studied to help with. I think the strongest evidence is for lactose intolerance. In fact, many people are, I don't know if you realize this, but yogurt actually has more lactose in it than milk does. Okay, so you actually, because when you make yogurt, back to yogurt again, but if you start with milk, you add back milk solids to make it thicker. And in that milk solids is milk protein that helps form the gel. but a lot of lactose. And so far more lactose than the culture needs to make the yogurt. So there's actually more lactose in yogurt than in milk. So, but these people with not severe lactose intolerance can eat yogurt just because the bacteria in the yogurt are very beneficial in digesting the lactose in your gut. And so it doesn't reach your colon where you get the problem. Another is prevention of diarrhea, such as with antibiotic treatment or Clostridium difficile infections, preventing these kind of things. These are probably the most strongest cases for benefit. And then a related topic are these prebiotics and symbiotics. And so prebiotics, we like to, I know it's always confusing, but prebiotics are these food ingredients that are not digestible, but fermentable. And they lead to a beneficial microbial community in your gut. Right? It's a simple definition. Examples would be like inulin, fructooligosaccharides, galactooligosaccharides, lactulose, which is like a metamucil. But galactooligosaccharides, fructooligosaccharides, and inulin, I'm sure these are all things that you eat now. You might not realize what products have them, but there's many products that have these in them. And then the last term I want to define is symbiotic. And these are what people refer to as products that have both pro and pre in the same product. And so this is just one example, not an endorsement, but just an example. The Stonyfield Farm smoothie, of course, I don't even know if this is still on the market. This is from a slide from a few years ago, but you can see in this recipe, we have inulin, right? So this has a prebiotic in it. This is actually going to help this drink be a little thicker. So it has a texture benefit as well, as in addition to being as fiber. And then they have the live active cultures as well. And you can notice that they don't say the names of any of these, right? This Acidophilus, Bifidobacteria, I mean, they name the species perhaps, but they don't list the strain name, right? So that gives you a little, this might not be a high amount of these probiotics in there. Okay. So in our gut, so there are 10 of the 14 organisms associated with our human body. In fact, in your colon, you have 10 of the 12 organisms, microorganisms per gram. Right? So it's an amazing number of organisms per gram. In fact, your stool is 50% bacteria, right, on a dry weight. Okay, so you are loaded, right? We don't, to get 10 to 12 organisms per mil or per gram in the laboratory is actually very challenging, right? We can't do that, right, without special fermenters and things. So this is an incredible high density of organisms there. Many of them not been cultured, or maybe they have, but they're very, in some cases they have, but they're very difficult to deal with. One of those studies we worked on, we've actually found there's over 1,000 different identifiable species in your gut, right? And the ones you have are going to be different than the ones somebody else has. It tends to be pretty stable. We did one study where we collected 3 samples from people in consecutive days, thereabouts, and they cluster very well together in terms of their community. So there's very little day-to-day movement. And that huge metabolic potential, of course, these organisms, so many of them, and they have such a diverse microbial or genomic gene content that they can do lots of things that we can't do and contribute to our health in that way. For the most part, as long as you don't enter a disease state, they're going to be pretty stable through your lifetime. At least that's the thought. And they're not just something you carry around, but actually are very important. We have, the Center for Nutrition, Learning, and Memory, I think it's right over here somewhere with the Abbott thing. And the gut-brain axis and how the gut bacteria communicate to the host, which communicates to the brain, that's all and it's a very hot topic now. So these organisms in the gut are not just something you just carry around. They actually, or just metabolize fiber. They're doing lots of other things for your health. In fact, animals that don't have any microbes in their gut, you can do this experimentally. You can take, for example, a pig and a pregnant sow and do deliver a piglet by C-section and kind of do all sort of sterile technique and move them to an isolator where no bacteria are there, and they live. And you do this with rodents as well, but they don't live as well, right? They have lots of problems, right? Having these microbes, they are very important for optimum health. Of course, in the mother's womb, they have no bacteria at least it's thought that they have no bacteria associated with them. And then so we have one project where we looked at the difference between C-section and vaginal delivery. There's a lot of interest in that because the birth canal and the birthing process, having witnessed, at least not experienced, but witnessed my wife's birthing processes. It's an amazing inoculation procedure. And so the babies are going to get a lot of organisms from the mom in this process, and that's believed to be very important. So they're normally colonized with things like E. coli, and Streptococcus, other facultative anaerobes. And then within a week or two, normally these kind of help make a more reduced environment that enable the more true anaerobic organisms to grow. And this is part of the reason why the infant formula companies are very interested. Breastfed babies tend to have much more bifidobacteria in their gut. And so that's part of the reason why we think they're so great, because breastfeeding is so great. And for formula-fed infants, at least traditionally, they have much lower abundance of bifidobacteria in their gut. But once they start eating solid food, this kind of whole situation changes. And then by the time they're age 2, they look like an adult population. We'll talk a little bit about that. Okay. So we're kind of getting into the first project. So it's always interesting when you get funded by infant formula companies. It's always, they're very pro breastfeeding, but they make infant formula. So it feels like there's always this dichotomy. But they truly want to make infant formulas that are better, that will for situations where mother's milk is not available or possible, that there's a product that delivers as close as possible the benefits of breastfeeding. But there's a lot of benefits of breastfeeding, such as fewer allergies. They have fewer infections, less asthma. There's lots of even like ties with obesity, perhaps, that have been tied with being breastfed versus bottle fed. So what is in human milk? So we're going to really focus on the human milk oligosaccharides from our perspective today. But we have lactose is 7%, so 70 grams per liter, a lot of lactose. Cow's milk is probably 50 to 60 grams per liter, so we have a little more. 4% lipid, just 8 grams per liter protein, so not very much protein actually. And then 5 to 15 grams per liter of human milk oligosaccharides. These are human milk oligosaccharides are things that are not digestible, right? So they make it right through the infant, and it gets to their colon where it can be fermented. And so it kind of depends on the situation. But essentially what HMOs are, they're lactose that's been decorated with lots of other sugars attached. And we'll go through that in a few slides. But it's just lactose with more stuff on it. With lactose as kind of the building block. And so they can have fucoses, or sialic acids, which is like an amino sugar attached to that lactose. And actually, it's an amazing diversity. So there's individual moms can have hundreds of these different structures there. This is like makes the analytical chemists this is like the dream for them, because they can study this for their lifetime and still learn new things, because it's just an amazing array of things that are there, and you keep learning. And so each mom is different. Okay, and then each mom is different during each. For example, today versus tomorrow, there could be changes. First baby, second baby, very early feed, like a colostrum, is going to be very different. The first feeding of an infant versus a baby that's 6 months, it kind of evolves as a moving target. So it's very hard. A lot of the work in HMOs is very difficult because to analyze for these things is very challenging, because a lot of times they're all the same composition but different linkages. Right? So it makes it hard to discern them by HPLC or something like that. Even if you get samples from the same mom over and over again, their composition is going to change, and so it makes it a difficult problem to study.
Does it change according to a plan, or is it just random?
Don't know. I think people, the breastfeeding advocates would tell you that it's nature's plan. I don't know if there's any evidence for that. But definitely at the beginning, there's a high push at the beginning, and it tapers off as they get older typically. And there's correlation with blood type, things like that too, that alter that composition. Yeah?
Is there the same amount of diversity of variation in cow's milk versus eating?
Oh, I didn't mention that. So good question. Cow's milk has almost none. we are very unique in this this angle. So as you get closer to us, they get more and more and more, but they might have, so we're in 15 grams per liter, they might be maybe in 5 milligrams per liter. And then the complexity of the ones that they have are actually, they just have kind of a few simple ones. And we have some, tetra and hexa and pentasaccharides that are very, you'll see, a little more complicated. And so that are not detectable in cow's milk. So that's part of the problem. So infant formula is not going to have any, or at least far less than what human milk will have. So the questions that we're going to talk about today are, what can we add to infant formula to make it more bifidogenic or kind of mimic what we see with breastfeeding? And it's going to be HMOs or HMO surrogates. How are these HMOs bifidogenic, meaning how they increase the amount of bifidobacteria in these breastfed infants? And then can we make these HMOs by using genetic engineering of E. coli? These are the 3 research topics we're going to talk about. Okay, so, all right. So the premise is that so in general, when we use prebiotics for other things, like when they add inulin to yogurt or things like this, it's believed, and there's actually lots, I don't know if you know George Fahey in Animal Science, but he's kind of the king of fiber, actually wrote the fiber guidelines for how much fiber we should all be eating. He's on the DRI panel that did that over in the Renaissance. But they do lots of studies where they feed people prebiotics or different animal species, and you can see measurable changes in the fermentation, the microbial population, different other aspects of gut health, or even cholesterol and other things that are important. And so things like the short-chain fatty acids, these are products of fermentation in the colon and are believed to be very important from a health perspective. And then a lot of times they use these prebiotics as sugar replacements. So polydextrose, we're going to talk about a lot. Polydextrose is actually used a lot in products because it kind of provides some texture. So when you start making like fat-free things or things with reduced sugar, like you imagine like a Coca-Cola has a lot of sugar in it. Well, you replace that with just a little bit of aspartame, right? You've really changed the texture of that product, right? Because now you've taken out a major bulking agent and replaced it with just a little bit of stuff, right? So some people don't like Diet Coke for that, or Diet sodas, because they have the mouthfeel you get from having all the sugar is different. So they don't use polydextrose in the sodas, but in other products, these are used a lot for those kind of purposes. And sometimes it's added just solely to be a fiber or prebiotic. So these are the 2 that we're going to talk about, the polydextrose and galactose, or galacto-oligosaccharides, or GOS. So the top one on top is PDX. And this is a sorbitol, mostly glucose, and a sorbitol and citric acid, like random polymer. And part of this makes it kind of interesting to study, but it's very difficult to analyze because every molecule is different in terms of what kind of linkages and stuff. And this is part of the reason why it functions as a fiber. It has so many funky linkages and stuff that we can't digest it. And it gets to the colon where the very dynamic microorganisms in our colon can have no trouble digesting this particular fiber. And then galacto-oligosaccharides is an interesting one. This is lactose. So we have glucose and galactose here. And then they add, using enzymes, this is normally an enzymatic process, they can add galactose onto this unit. And so you get a product with different degrees of polymerization. 2 would be just lactose to maybe DP8 might be the maximum you might see depending on the process. So they have a range of these polymers. And this linkage is not digestible by us, so it makes it to the colon where it could be fermented. Okay. Like I already said this, they're indigestible, fermented into the colon. And some, with adult studies, in animal studies, they promote the growth of lactobacilli and bifidobacteria in the colon. So you can see increases in these, and presumably that's good. And so the premise of the project that got us funding for this was, can we add PDX and GOS infant formula, and would that be beneficial? To add things to infant formula is actually a big problem. You can't, when you want to add to food, once it's approved as food grade, it's very easy to add to foods. Infant formula has another threshold it has to meet for it to be approved for use. It's not a drug, but it's not food, somewhere in between. And I don't fully understand it, but it's more complicated things approved for them to use. It makes sense, though, if you think about it. An infant, all their nutrition is one thing, right? If they're drinking formula, like when they're 3 months old, 100% of their diet is that infant formula, right? So if there's any kind of bad thing about that, that's going to be even worse, right, than it would be for you who just drinks a little bit every once in a while, right? So it's very important for infants to really get it right when you tinker with infant formula and try to improve it. So the objective of this study was to investigate the effect of pediatrics and GOS on changes in short-chain fatty acids, which is products of fermentation, the gut histomorphology, and the bacterial community in using piglets. When I actually came to U of I never had worked with piglets before, so it's been quite fun learning how to use piglets as a model. I've done several studies now with piglets. The advantage of using piglets is that their GI tract is much more similar to a human infant's compared to like a rodent. They ferment in a colon, for example. Rats don't ferment in their colon, they ferment in the cecum. Their GI tract is just different. Okay? So it's much more similar to us. So that's a big advantage. They're cute little guys too, which is always kind of troubling, but fun to work with. They grow really fast. So we only have them for 3 weeks, and then they're weaned. And they go from little guys, like puppy, little guys, maybe 2-3 pounds, to just being, grab around the belly to pick them up. They grow unbelievably fast. So what they accomplish in 3 weeks, the growth they accomplish in 3 weeks is about what a human infant would do in 6 months. So it really accelerated growth with them. And so we had 3 groups in this study. We had a sow-reared group. So these are piglets that stay with their mom. A mom actually has litters of anywhere, well, it depends on the breed of pigs, but the pigs they have at South Farms, I don't know if you guys ever get down like First Street towards like Curtis, south of Windsor. There's a pig farm right there. Campus Porcine Research Center. I guess it's Swine Research Center is that one. They provide pigs for studies like in Irmal, which is right next to my building, and other places. And so those sows have anywhere from 10 to 15, maybe even more, 18 sometimes, piglets per birthing cycle. So they're very prodigious, lots of little piglets. And we have, so the sow reared that stayed with the mom. We have formula-fed piglets that got a formula without any prebiotics, and then we have one with prebiotics. And these are at 2 grams per liter, which is the concentration that they want. That's the goal for the infant formula. Yes?
This is a little bit off topic, but why exactly do pigs grow so much faster? Do they just have more growth hormone?
No, that's just their biology. It's actually kind of like we always just joke, oh, they're pigs, They eat what you give them. They eat a lot. They're always hungry. So I think that's just, just part of being a pig. That's the best I can tell you. As a microbiologist, the best I can tell you. But it's very useful for us because as a model, you don't have to do a 6-month experiment. It can be a 3-week experiment. But there's a lot of work. We have them in individual cages, and we have a whole system of how formulas. We have an engineer actually help us design how the formula is delivered about every hour or so, a set amount, and we can actually know how much each piglet is getting and how much they're eating, and it's a well-controlled kind of nutrition study. Then we have, these all piglets came from 3 different moms, and they get shuffled, randomized, and then they each get assigned to each group. So the sow group came from 3 different sows, but they all nursed on the same sow, which is they were all sisters in there. Pigs are good with that. Learn all sorts of things. All right, so this stuff was less interesting, at least to me, so I suspect to you as well. So I ran through this stuff with the total weight gain, intestinal weight, intestinal total length, ileum, which is the end of the small intestine, and ascending colon, dry matter. That indicates of water. So if they have a little diarrhea type or very loose, situation in there, then you'll see differences in the amount of water content. And so fiber, like these prebiotics, are, if you give too much of that, they're going to have a little problem in that regard. But from it, really no effect or very minor effects with with these kind of more basic analyses. And then, so we did a whole slew of HPLC looking at these fermentation products, and there's very little difference. And where there is a difference, it's between the sow and the other two groups. So the formula plus prebiotics are very similar, and the sow is somewhere different. This is kind of the conclusion from this. All right, and then we get to the micro part, which is more my interest. But we did some work looking at the lactobacilli and bifidobacteria population from these piglets, but also did some high throughput sequencing analysis, which was fun. This was our first time trying to do that. So we've gotten a lot better at it now. But one way you can look at the microbial population, just look at the diversity that's present in that those samples. So this is AC's ascending colon, which is more the beginning of the colon, I would say. And the ileum was the end of the small intestine. So the ascending colon, the colon is where you have a lot of fermentation, especially the ascending colon. And the ileum is where fermentation just kind of starts. Upstream of that is where food goes through really fast. There's not really much action from bacteria because we're taking everything we can from it, like the host. And by the time it gets to the ileum, that's where the microbes now have a chance to go after it. And so in this case, you can see in terms of diversity, the sow group, and the use in the ileum was much higher. So I can use my pointer here than the other two groups. But in the ascending colon, where there's much more diversity and much more fermentation happening, the three groups are very similar. And we can even go through lots of these things, but I think the point, this is some analysis from the ileum, and the next slide is from the colon. And so you can look at these are just presenting the genera. So these are things like Lactobacillus or Escherichia or Clostridium, for example, that had a treatment effect. And what you can see is that most of the time, the red and green are very close, and the blue is different. Sometimes the blue is higher, sometimes the blue's higher, sometimes the blue's lower. Very rarely is the red and green that different. It's really the sow that's different than the other two groups. In the colon, we see the same thing. We see a lot of things like this where these are very low and sow is high. Of course, Catabacter, of course, is one of the ones that was kind of unique. But Tannerella, very high in sow, low in these groups. And it was very Fecalibacterium, very high in the formula groups, but low in the sour groups. Lots of these kind of things. This is a problem in our field, actually. So we could do this analysis, but then we don't know what to say. Because now it's like, wow, there's all these differences. Is that good? I don't know, but it's different. That's kind of where we're at. This kind of thing. The understanding of it is not as fast as the technology to analyze it. So if you look at that community and condense it down to like a dot on a two-dimensional plot, okay, which is a challenge, using a process called unifrac. Each one of these dots indicates a different piglet, or at least a sample. Each piglet had one ileum sample and one ascending colon sample. So what can we learn from this? Well, first thing, here's the ileum from the sow, and here are the two ileum groups from the formula plus prebiotics. So the formula and prebiotic groups are very similar to each other and indistinguishable and very different from the sow rear group. And over here is the colon. Here's the green is the ascending colon from the sow rear group. And then you can't even tell, but here's the kind of the mustard and pink color of the two formula groups in the colon that are overlapping, totally overlapping. And on top of this, you can see a lot of those bacterial groups that were on the previous slides are on here, listing, pointing at the different groups that were indicative of that particular treatment. Okay. So what did we learn from all that? So the prebiotic supplementation was not harmful. They still grew well. Their intestines were no different than the formula control, that everything seemed okay with that. We saw large differences between the sour root group and the other two groups, and very little differences between the formula prebiotic group. And so that's actually quite interesting. I could tell you a little bit about why that is. That's not really a flaw. It's just kind of, they're really not good control. The sour piglets live with their mom. They're in a room in the basement of Ermel, but they're with their mom. What does that mean? Well, there's poop everywhere, right? And they're licking their mom, licking each other. They're a lot more, just a different experience. The formula-fed piglets are in individual cages. They can't touch each other. They're kind of, they can see other piglets, but they can't touch each other. They can, and they deliver this formula, through this tube in a bowl that they lap up and make this lapping sound that's very entertaining. And they, very lip smacking, but it's a very different experience, right? So not necessarily getting our formula to look like the cell group is perhaps impossible with that kind of model. So we're trying to actually nurse, and some of our collaborators, like Dr. Dom, is actually trying to develop a system to milk pigs, collect the milk, and then use that milk to deliver through the system, and then you can have an actually true control where you can compare sow milk to formula in the same, otherwise same system. Yeah?
Is it possible that the sow milk composition would change in reaction to being like mechanically milked versus like milk?
So that's part of what I was talking last night about raw milk versus pasteurized milk, but that's an issue. So how you collect the milk and how fast you chill it and how fast you get it to the baby pigs, right? Because when you milk a cow for, like, you got me going. So cows were not bred to give us milk that we store for a week in the refrigerator, right? They make milk for the calf, who never stores it. He or she eats the milk, gets it from the teat, and then that's it. There's no storage, right? No storage outside the animal. We've come along and figured out, we can milk that and we can use that milk. But it's not, that milk wasn't designed to be outside the animal and not treated in some way and be stored for periods of time, right? It should be pasteurized or at least properly chilled and used properly.
I mean more in the sense that the sow would the sow like release different hormones?
That's another issue. Yeah, how would the milk composition change by adding that stress to the pig sow? Unknown. Nobody's really milked pigs before, so it's not really. These are things that will be addressed or asked, but it's entirely possible because that's the stress. Stress definitely impacts milk composition and microbiota composition, things like that. And that could be a part of why we see our difference too, because they're an individual. They're kind of they would prefer to be with their friends and with their mom and kind of running around with that rather than being these individual cages. So some would say that's maybe a stress just doing that, and that could be part of our problem as well. So I kind of did a little bashing of the model, but in fact, this model, though, even with that, is still far better than doing this like in rodents, right? Which would be even worse. So this is the best option. Actually, here in Illinois, I don't know if you guys realize this, we do an unbelievable amount of pig biomedical type research here on campus. Definitely in ACEs, but people outside ACEs as well use piglets and pigs as a biomedical model. So, in fact, the University of Illinois, not UIUC, but the University of Illinois' overall research director, I don't know what his title is, but Larry Shook is famous for being sequencing the pig genome about 5 or 6 years ago. Now he's the director of kind of the VCR for the whole university. So I don't know what that title is, but okay. So that was all nice, but we wanted to move towards something that was more similar to what was in breast milk and used in HMOs, human milk oligosaccharides. I already said all this already, but just to remember, these are 5 to 15 grams per liter in breast milk. Remember, we only did, for the pre-box, we only did 2 grams per liter for each one. So we were at 4 grams total for those two, the piglet study. And all those decisions are actually made by cost-benefit analysis, right? We could add, okay, how much can we add that would be great? How much can we add that would be affordable, right? We can't make something that people can't buy because it's just way out of bounds, right? So they want to get the most bang for their buck with these ingredients. Okay, so oligosaccharides start off with these main building blocks. Glucose, galactose, of course, these are the two saccharide sugars and lactose. Glicnac, or N-acetylglucosamine. Then we have fucose and sialic acid as the other two. So we have lactose, which is a beta-1,4 linkage of glucose and galactose. Sure. Got some sugar chemistry here. This would be a very simple HMO where we have lactose with a sialic acid attached. It can be either 2,3 or 2,6 linkage typically, the two that are seen. So this would be what we would call 3-sialolactose or 6-sialolactose, depending on which linkage. So there's the 3 and 6 SL. But we can also decorate with fucose, as indicated by a triangle, and this can give us things like 2 and 3 fucosyllolactose. These are probably the more abundant of the human milk oligosaccharides and most simple. But we also have ones where we have lactose. And then we have a lactosamine, which is lactose, but the glucose is replaced with glucose, glicnac. And this could be different lengths, so these can even have multiple units of that attached, of course, to different linkages. And then they can be further decorated with fucoses and sialic acids. So leading to why there's 200 or more known species or individual molecules of human milk oligosaccharides have been detected. So I pulled this from one of Dr. Donovan's students' thesis, because I haven't found this anywhere else, but this is they took several different studies and kind of figured out what the average concentration of some of these HMOs are in human milk. And so LNNT is the simplest tetrasaccharide. Can be even this is probably the most abundant of those. We have 3 and 6 SL and 2FL and 3FL. And you can see 2FL is very, very high. So maybe half of the HMOs in breast milk can be 2FL. And we have some the individual sugars can also be detected. Okay. So we've been working on how confirming that bifidobacteria can ferment these human milk oligosaccharides. At least that's been the premise, and we've been able to confirm that. And then understanding a little bit about how they do that. And so to do this this first part's very straightforward. We have a growth media that is defined that we can add only the carbohydrate of interest. And if it can't ferment that carbohydrate, then it doesn't grow. If it can't ferment that carbohydrate, then it grows. Right? So we can kind of do these little growth curves in our bioscreen C. It's kind of a fancy spectrophotometer that's in our anaerobic chamber in Bevere Hall. Dr. Lou's been in there before. And this is kind of an example of some of the growth curves you can get from that. Maybe that was me. Have I gone long?
No.
Okay. Let me know if I need to wrap it up. I think I'm on time. But anyway, so we have 3SL. And we see none of the lactobacilli can ferment 3SL. And the two Infantis strains, this one here and this one down here, this is actually a commercial strain that's in some products. This can ferment 3SL. If you look at 2FL, none of the lactobacilli could do it on the left, but the same two bifidobacteria are pretty well fermenting this. This is just kind of a summary slide. And the LNNT, we see some additional, the two Infantis strains do well, and then there's another Breve strain that does well. And then there's some lactobacilli can actually ferment LNNT. And so we've been looking into that in more detail. So the conclusion from this study, at least I kind of abbreviated it, is only B Infantis can ferment all these HMOs that we tested. And this actually, how that got that name, right, is isolated from infants, right? That's very clever with B Infantis. And so it is actually very good. And so what people find when a human infant is breastfed, if they don't have B Infantis in their gut, all the HMOs go right through, and you can detect 95% of them in their diaper and stool samples. And so, but once they get colonized with B. infantis, then there's none in the stool. It all gets fermented in their colon. So it's a very important organism for fermenting these things. I didn't present any of this data, but Enterobacteriaceae like E. coli, Klebsiella, Enterobacter, some other fun organisms, and a lot of them pathogens aren't able to ferment these things. This is a very unique property of Bifidobacteria, and specifically B. infantis that's very good at this. So just kind of from a review, but just kind of summarize some of our data as well, that these human milk oligosaccharides, with B. infantis at least, it takes in these sugars whole, because we never detect them being chopped up on the outside and then taken in individual pieces. It actually takes up the piece whole and then metabolizes it inside the cell. This limits the size of the HMO that it can digest, so it doesn't do very well in the really big ones. Breve, the one that grew on LNT, that actually is very good at secreting lots of these glycosidases and chews them up into little pieces and it takes in the little pieces. So it's two different approaches. What the significance of that in the gut is unknown. The thought is, this is a very selfish approach, right? You take the whole sugar, it's mine, I'm taking it inside, and then I'm using it all. Whereas Breve is more of I'm cutting it up in little pieces and taking what I can, but anybody else who's out here can take it too. Right, so there'd be some interesting aspects to that. Okay. And the last part, I probably spend the most time, I think it's perhaps the most interesting to you guys, is the synthesis of HMOs. And so we're going to talk about 2FL the most. So 2FL. 2FL is a major bummer. Actually, HMOs are a major bummer in that you think of all these fun experiments you'd like to do, but you can't, because you can't buy them. They're $2,500 for 100 milligrams of 2FL. And remember, it could be 2 or 3 grams per liter in breast milk. Pigs, right, the piglets, if we want to do a piglet study, they do at least a liter a day. Right, so you would need, for one piglet, right, and you want a similar concentration that you'd have in human milk, right, you're talking tens of thousands of dollars per day per piglet. So it's not, not even in the cards, so nobody does that. Yes?
So is this being harvested from human milk, or is it being made?
Mostly, yes. So there's two paths of people that supply it. But you can imagine human milk's very difficult first. Okay, first you have to get human milk, which is possible. But then you have to separate it out from all these very closely related ones and sell just the 2FL, and you're going to lose a lot of it by purifying it away from the 6FL and these other things. So it's an analytical problem that I certainly don't have an expertise in, but it must be hard to do because it's very expensive. There's a chemical way to make it, but it requires GDP fucose. And an enzyme transfers the fucose from GDP fucose onto the lactose. But GDP fucose is crazy expensive. So it's the same price, actually, so it doesn't help you. So some people, and we'll get into that, have been trying to make more GDP fucose, and for that purpose. But we've kind of combined approaches, so you'll see. Okay. I keep hitting this button. So what is it? This is the lactose, again, with this 2 linkage here with to the fucose. So it's L-fucose, galactose, and glucose. It's normally found, actually, about 80 to 90% of moms make a lot of this, and then 10 to 20 make none. Right? So there's secretors and non-secretors that they're called. And so the 10 or 20% that don't make 2FL are there. And so depending if you collect milk from just one mom, for example, and that mom's a non-secretor, you could be purifying forever and never find it, not be very successful for 2FL. But for the ones who are secretors, they actually make a lot. So there's really 2 different groups. And there's people trying to figure out what the significance of that, health-wise. Bless you. But it can count for 38, maybe even 40 or 50% of the total HMOs in moms that at least that make 2FL. And so from other work we knew B. infantis was very good at fermenting it and could grow very well, very fast with that, actually almost as fast as growing on just glucose. Okay, and so the next part, we'll talk about this some more, but moms that are secretors, their babies get less Campylobacter jejuni infections. moms who are not secretors, so have very low 2FL and other fucosylated sugars in their milk, will their infants have more Campylobacter infections. Campylobacter jejuni is either number 1 or number 2, depending on the study, kind of right up there with Salmonella in terms of frequency of illness, foodborne illness in the U.S. In fact, 50% of the raw chicken you buy at the grocery store has Campylobacter on it. So our USDA project actually is all around trying to figure out ways to get Campylobacter jejuni not colonizing the chicken, because if the chicken doesn't care, it's a commensal organism for them. It just gives us problems, but the bird doesn't care. No issue for the bird. So, so these things, these sugars look like the same sugars that are in your mucus. Mucus is actually glycoprotein. It's a protein that's heavily cross-linked and heavily glycosylated, lots of sugars on it. And these human milk oligosaccharides are the same sugars that are what is attached to the mucus. These sugars on the mucus are what pathogens use to say, grab on, right? With these lectins, carbohydrate binding proteins grab onto these sugars, and that's where they grab on, colonize, and kind of colonize the mucus and live in our gut and initiate the problems, right? Or the commensals used to anchor in and stay in our gut. Perfect, just in time for me to walk back. Okay, I think I'm just drying off. All right. So it has some interesting applications in terms of preventing Campylobacter and disease and other diarrheal illnesses. So how are we making 2FL? So this is a collaboration with Yangsu Jin. We take advantage, this part here in 1 is the GDP fucose biosynthetic pathway. So this is the GDP fucose that can be transferred onto lactose by this enzyme. I make sure I don't say it because sometimes I say inappropriate things, but it's FUCT2, right? That enzyme, or fucosyl-transferase we'll call it. It's actually an enzyme. This pathway is this FUCK, which is also even worse, more troublesome. But we have this pathway. So this pathway actually exists in the E. coli. They use this to make GDP fucose that they use to. There's actually some fucose in their cell wall in a peptidoglycan. So they make a little bit of this, but not very much. And so what we've done is increase the expression of this pathway, this MAM-B, C, WCAJ, and GMD, and then E. coli. And then we've also introduced this enzyme from fucosyl-transferase from Helicobacter pylori. And so what people had done in the past had done just step 1 and been able to make some increased amounts of GDP fucose in E. coli, which then they subsequently purified and then used purified enzyme to make 2FL. Right, but that's not as convenient as having E. coli do it all for you in one time. So we have a batch and a fed batch fermentation. So this is the batch, and we have, let's see, we'll do lactose first. Lactose this is a 15 gram per liter fermentation. You see the lactose goes down, and as lactose goes down, we can see the optical density, which is in green, goes up. So the cell number in our fermenter increases, and the amount of 2FL goes up. We can get all the way up to a gram per liter, which is approaching the concentration in breast milk, right? So that's good news. And there's not making any of the other ones, so we don't have to separate it out like we would for breast milk. And by doing a batch, fed batch type, so this is, you add lactose, let it consume it, add more lactose, let it consume it, add more, and so you kind of keep doing this it's called a fed batch. Actually, if any of us are ever interested, in my food micro class I teach with Yangsu Jin, we have a whole lecture on fermenter design and fermenter strategies like batch and fed batch fermentations if you guys are interested. But you can see by doing that, we can get all the way up to 4 grams or more grams per liter of 2FL, which was super exciting for us. You know, just 1 liter of this, for us to get that 4 grams, if we had to buy it, right, that's in the $50,000 or something for us to buy that much 2FL. It's not really plausible. And some analytical work to hopefully prove to you that just like from MS, we can see that our standard and the 2FL that we are making by our E. coli is the same. We've actually done NMR as well. and some other Maldi-Toff and things like that, and it's definitely 2FL that we're making. And so what we've been doing to kind of increase this situation is we're now working on, this is, just met with a student yesterday about this, but she's doing a high cell density culture. So these are going to be an OD, we're hoping to get to an OD 200, which is like a paste, right? This is going to be a very dense bacterial culture. You do that by feeding them low glucose at the beginning. That's enough to let them to kind of grow and increase in numbers. And at some time point, you're going to add IPTG. This is going to induce the expression of your pathway, the enzymes you put into E. coli on the plasmid. So to make more GDP fucose and start making the fucosyl transferase. And after you give it some time for that induction to result in those enzymes being made, you can add lactose, and you see and then lactose be consumed and converted to 2FL. The premise is the E. coli strain we use is actually a non-lactose fermenter. So it's not using the lactose to grow, it's using lactose just as a substrate for this enzyme. But it's not perfect, as you can imagine. If you closely look at this, right? We have 15 grams of lactose, but we're only making 1.2 grams of 2FL, right? And molar-wise, that's even worse than that, right? Because that includes the fucose been added onto it. So this is, we're losing 90% or more of this is gone, right? And it's used for growth, even though this is why this takes forever, right? This is a very long fermentation. Yes?
Have you considered doing sort of like computational approach to see how you could increase the yield of the 2FL? Or?
What do you, like what?
So just modeling the pathway and seeing if you can fine-tune any of the...
We have a little. Dr. Jen does that a lot with the biofuel stuff. And that's, for this project we only have one student. So it's not as army as we have the biofuel projects. But we have a little. And that is, I know for his other projects, I'm on like all student committees, so I can have a full kind of good grasp of what they're doing. But they have, they can make significant improvements by fine-tuning the expression of each gene, figure out where the bottlenecks are, and kind of optimizing the expression. We haven't, for me, just getting 4 grams per liter. Now it's like, okay, I don't care if we get any better. Let's do a couple liters, and then I have enough to do what I want. But Dr. Jen, and the student, they want to make more and more, make it better and better and better. Like that's everybody has a different interest. So I'm less interested in the synthesis and more interested in the using it. But it's still fun. It's a great opportunity for me. And here's I already showed this. And we actually we have a collaborator at USDA. Neil Price is a carbohydrate glycochemist. It's been super helpful. And we were banging our head for a long time before we met him. And it's been just revolutionary for us. One of the things he helped us with this purification strategy. So this is just activated carbon. And we could pour our 2FL, our E. coli growth media through this crude column and just with some vacuum suck through stuff, and it's eluted with ethanol. So the lower ethanol content you get, this is what the flow through looks like. This is kind of the spent LB media, this dark brown. And this here is with the ethanol extract where it has our 2FL here. And it's very simple and actually ends up being very clean. I'll keep hitting that button. So this is a Maldi-Toff of our purified 2FL, and this takes us like an hour we can do a liter. This is a very fast kind of process. And that's just, it could be faster if we just had a bigger scale. But there's the Maldi-Toff of the purified 2FL that we have, and you can see that these are just different salts of the 2FL, and so these are the only 2 significant peaks in that. So it's a very easy purification. We can separate that 2FL out from the rest of that fermentate very easily. So we're able to make significant quantities of 2FL, and this is actually in the literature where we have one manuscript already, and we're working on another one. We are far in front of any of the other published literature in terms of making these kinds of sugars, which is great. Purification strategy is working well, and so one of the goals for the student is to actually increase rate, increase yield, and one of the ways is to optimize the pathway. We're also trying to inhibit the ability of E. coli to use fucose in what it normally would use it for, so it directed more. We're still trying to figure out what's happening with lactose, because the strain shouldn't be fermenting lactose, but it does a little bit, and there's this whole lactose part that we're still kind of confused by. We're trying to figure out. So what are we going to do with this 2FL now that we have it? So I kind of already talked a lot about this, but C. jejuni, major cause for overnosis in the millions of cases per year just in the US. It's actually been associated with Guillain-Barré syndrome. And I don't know if you guys know Guillain-Barré syndrome, but this is a very terrible paralytic disease. And what happens is, the premise is people that get most of the people, the vast majority of people with this syndrome have had a previous Campylobacter infection. And what they notice is that some of the antigens on the surface of Campylobacter mimic the antigens on some of the nerve cells. And so if they develop inappropriate antibodies for this Campylobacter that also recognize themselves, they develop this autoimmune disease sometime later. Right? So it's Campylobacter is bad just from a diarrhea perspective, but there's actually some long-term consequences for a significant percentage of those folks as well. And so we want to use these fucosylated glycans. We're actually going to feed it to chickens next month. Actually, we're doing our first chicken. This is kind of exciting because I've never done work with chickens either. Before coming to U of I, actually, I'd never done anything with animals. And so I've done rodents, pigs, and now chickens. So it's just kind of fun being over the college ACEs and lots of collaborators who know how to do all these things. And we're going to be feeding 2FL to the chickens, and it's a modified 2FL to prevent the chickens from being colonized with C. jejuni. We're actually using 2FL and using that as a hook to identify lactobacilli that have great affinity for it, and in hopes that we can identify novel probiotics for chickens that you could feed to the chicken, and they would occupy that niche before Campylobacter get exposed to Campylobacter. There's actually a lot of commercial products for probiotics for chickens that people use for these kind of purpose, but ours is the first one that's going to be more mechanistically designed. And then I was, of course, the end here, but Jenny was with the Piglet study. Toxwan was doing the Bifidobacter fermentation, and then Panchali is the one I co-advised with Dr. Dennis dude that's making it 2FL. And so all the work I talked about today was funded by Abbott, Nutrition, or Mead Johnson, or USDA. And then these are just the current group. So we have several grad students now, and one postdoc and a lab tech. So that's it. So I am glad to take any questions from you guys. I'll be here. I'm staying for lunch too, so I don't miss a lunch typically. So obviously, so I'll be here to answer questions then too.
Any questions?
You guys asked why we went. Yes.
Is 2FL in the human made in the epithelial cells of the human or in bacteria?
Oh, it's all made by the host.
So
It's made in the milk ducts.
milk ducts. It's epithelial cells, probably.
Yeah.
I mean, so so for women that don't produce this, it's got to be in their genes.
Yes.
It's just not programmed in the.
Right. So this relates to their secretor status, and that has applications to other things beyond just whether they make fucosylated glycans. It's like a blood type thing and things like this. So there's some. Yes. So they make it in the mammary gland, but they also make it in their gut. Specialized cells in their gut make it for the mucus as well, and the goblet cells.
So if they don't have it, do they sense that and supplement, or is that not going on?
That's I think in some respects the future. If we understand what these different compositions, what these individual ones are doing, and we find out like some moms are making ones or the other, is that what's best for their infant, or is there maybe a supplementation that'd be better, or that's
Just not knowing.
that's no. And the big problem is because you can't buy these things in appreciable amounts to do these kinds of studies, and so Abbott Nutrition has, as far as I can gather, an amazing number of patent coverage in this area. This is why part of a lot of the Center for Nutrition Learning and Memory work is related to HMOs and memory. I think, at least my interpretation is they have a lot of interest in that area, right? From a scientific perspective, but also from a cash money perspective.
I have a question.
Yes.
Have you ever think of transplanting the 2FL pathway into lactic bacteria?
So it make it in the gut?
Yeah, or like somehow in the milk.
Yeah.
Like that. So that you don't have to purify, right?
Yeah, that would be nice. The trouble is E. coli is so easy. It makes such high amounts for us, and it's so easy to work with. We have all the tools and stuff. It's just much more. Yeah, certainly as a first step.
Okay.
I think for next steps, that would be interesting to try. If we could do that, then you wouldn't have to purify. You could just feed it,
That's right.
and maybe it would make it in yogurt, for example, while the milk's while they're in fermentation.
Sure.
Yeah, that would be cool.
More questions?
Yes.
It's more of like a general question, but I've heard like claims where they say that probiotics that are marketed, in yogurt and stuff that are that claim to have health benefits don't really have those health benefits.
Okay.
Have you heard that claim, and is that sort of like a fringe view, or is that a different sense to that?
Yes and no. So, okay, so here's a problem. You could say you have probiotics in your food. You don't need anything else, right? You just say, we added probiotics. Okay. And so there's a lot of I would say kind of shoddy products that are kind of trying to jump on the bandwagon without any kind of justification for what they're doing, any documentation for the amount that they're using. And if they do have some kind of data, it's pretty, right, not that good. But just enough to say they have studies or something. Like they did 5 people ate it for a week and that's it, this kind of thing. And so, but the ones, there are several that are very well studied, lots of NIH-funded studies and commercial studies, so things like BB12 and NCFM, the one in Activia yogurt, for example, that's been heavily studied. And so they get into trouble because so Activia got sued. I don't know if you know, Dana got a huge lawsuit in that front. So there's lawyers who just look for people that make claims on their product and say, I think we can win that one, and they just fight it. Not because, and so they end up settling that lawsuit. So they adjust their language on the. You and I probably wouldn't notice the difference in the language that they use, but from a lawyer perspective, it's suggestive versus a little more than suggestive, perhaps. But you have to kind of really get into it to kind of sense these subtleties. But they had some studies, but I don't know. When I first came here, actually, somebody from the Today Show called and asked, would I like to come on and talk about Activia yogurt and whether that's any different than regular yogurt, and do this little spiel. And I was like, ooh, I don't know. You're gonna have to tell me a little more about what you plan, because first I'm not an epidemiologist. I don't feel very comfortable evaluating clinical studies because that's not really my expertise. But if you're going to tell me that this bacteria, like eating yogurt. Okay, first, for example, E. coli 01578H7. This is the E. coli that causes all sorts of problems, right? Very dangerous organism, right? And then we have E. coli in our guts, millions of them per gram, no problem, right? Most E. coli, no problem. Few E. coli, big problem, right? They're all the same species, right? They're different. Why is it so hard to believe that different lactobacilli or bifidobacteria can be different? The genetic content is actually within the species, far different than it is between us. We're all different, with different disease, hair colors, skin colors, eye colors, all sorts. We have all sorts of disease tendencies, whatever. We're all different. Why can't they be different? So it's important. This in our field, what we say, the important thing is if you want to say you have some kind of claim, you have to test your strain and your product at the amount that you're going to have it. And then if you do that, then you can claim that your product with that thing, that probiotic, has some kind of whatever benefit that you're testing. But it is not appropriate to say any yogurt would do that. It's possible, but they didn't test any yogurt. They tested their yogurt. But I ended up declining because the problem is everybody they asked gets money from somewhere. They made them like, I can't do that. And I was like, I was too new. I was like, I don't really know. That seems like I would take me down a path that would not make me very popular, so I don't want to do that. And then what I wanted to say, that they didn't go with what they wanted, so they found somebody else, which is fine. Better for everybody.
Okay, more questions? So let's take that.
I'll have more long-winded answers maybe during lunch.
You can ask them during the lunch. All
right.
Thanks to Doug Miller.
All right, thank you.